Hello, my name is Clint Spiegel. I'm a professor of biochemistry in the Department of Chemistry at Western Washington University. One of the primary focuses of our research is the study of proteins involved in blood coagulation. The process of properly forming a blood clot is rapid and highly regulated, and when it does not work properly, various life-threatening disease states can arise. For example, one of the proteins involved in blood coagulation is called factor VIII, also known as anti-hemophilic factor. Defects in the gene that encodes factor VIII result in Hemophilia A, an X-linked recessive disease that affects 1 in 5,000 males worldwide. Hemophilia A symptoms consist of both internal and external bleeding and can affect joints, muscles, digestive system, and brain. The most effective treatment for Hemophilia A is called factor VIII replacement therapy, where a patient receives an infusion of recombinant version of functional factor VIII. While this treatment is quite effective in correcting blood clotting, there are significant clinical complications that arise from it. About 30% of patients receiving replacement therapy develop an immune response to the infusion, leaving the recombinant factor VIII ineffective. Our research is aimed at trying to understand the nature of this immune response to factor VIII replacement therapy. In collaboration with hematologists at Emory University Children's Hospital in Atlanta, Georgia, we use biochemical techniques and structural biology approaches to understand how antibodies recognize factor VIII at the atomic level. Our research projects begin with the production and purification of recombinant proteins to form complexes between domains of factor VIII and their associated inhibitory antibodies. Our recombinant proteins are produced in a bacterial model system and subsequently purified through column chromatography. Once highly pure protein complexes are formed, we use a technique called X-ray crystallography to study the detailed atomic structure of each factor VIII antibody complex. First, we attempt to coax these protein complexes into crystals that are often too small to see with the naked eye. Once crystals have been grown, we mount them with a loupe, flash freeze the crystals in liquid nitrogen, and subject them to a high flux of X-rays from which we can study their diffraction patterns. Once diffraction data are collected, we can generate an image of the electron density for each atom within the protein complex. With the use of modeling software, we try to solve this complicated three-dimensional puzzle by building atoms into the electron density, which will ultimately lead to the complete characterization of the protein structure. Through understanding the detailed nature of how factor VIII is recognized by inhibitory antibodies, we hope to develop more active factor VIII therapies that can evade the immune response associated with factor VIII replacement therapy. Thanks.